Eva Dyer from UPenn.
All right. Thank you so much for the invitation. It's really exciting to be here. So gonna kind of switch it up a little bit, which has been a theme of the workshop, very interdisciplinary. It's been exciting. So my lab, we work a lot on representation learning, specifically for time series data, and most of the applications are applied to different neural recordings from the brain as well as the body, so behavior as well. And what I'll talk to you about today has been some recent work that we've been doing over the past few years, inspired perhaps by the recent advances in a lot of generative AI, and in particular, wanting to harness the amount of scale of data out there and to see whether or not we can build better models to interpret brain activity by scaling up our computation and data. So in terms of the motivation of this work, I think we all can appreciate the complexity of the brain. It's a highly distributed system. And in order to start to, get insights into how this very complex and distributed system works, in neuroscience, we want to try to come at it with a certain level of control, right? And what does that look like? We're going to use a particular task or underlying behavior to be able to probe a particular circuit or region of interest, and often because of just constraints in how we can actually record from the brain and individual neurons in the brain, we're just going to see just a snapshot of the full activity that might be present across the full extent of the brain. And then also just due to constraints in actually recording technologies, we're often left with a relatively small number of individuals or animals that we can actually record from. So what this means is that each neural recording really just gives us this snapshot into the full complexity of this system. And so we've been really excited by the idea of maybe taking all these somewhat fragmented neural data sets and putting them into one unified model, which might be able to get something that's greater than the sum of its parts. And so really for us, this is like, we can kind of imagine that each data set is just giving us one piece of this patchwork, and really our goal is to somehow algorithmically stitch together these different sources of information into this model of hopefully cognition in the future. And so to do that, or what might this look like, especially for those of you that haven't necessarily had a lot of exposure to different electrophysiology recordings. So this is kind of the state of the art in terms of what we can actually probe in terms of neural function. So these are depth electrophysiology probes that can be inserted into the brain and often span multiple different regions of underlying neural circuits. And so now if we imagine taking some of these really large open source data sets, in this case is coming from the International Brain Laboratory. These are 700 of these probes that are spanning different parts of the neural circuit across 140 different mice. And so now if we imagine just, what would this look like to kind of put this all together, we start to see that instead of just seeing this small little swath of tissue, we can imagine that we could actually, span most of the hemisphere of a brain and get a very dense sampling, but only through this sort of aggregation step over many animals and many different data sets. So this would be kind of like the vision of the future if we could do this effectively. And also just to set the stage, what could we do with this potentially? We could imagine that, if I could record from just one probe in one animal, but potentially predict what was happening in many other regions of the brain, I would have a much more holistic picture of how, multi-regions are communicating and what are the underlying computations they're in. So this could be like this kind of vision of this predictive model that can fill in the gaps, given just a small subset of recordings. So, this is a really exciting goal and an idea to be able to aggregate all these data sets, but, there's been a number of challenges that have made this, a hard problem. So one is just the fact that all of these data sets are very heterogeneous, so they'll be probing decision-making or visual processing or motor and action planning. And so then the underlying, stimuli or inputs coming into each of these data sets will have different distributions and underlying properties. And the data sets, of course, if I'm studying motor planning, maybe I'm only measuring in motor cortex, but maybe there's other regions that, that could also be, of course, important in this. So the first is just the data sets have very different underlying distributions and inputs coming in. Kind of even more of a technical challenge that has to do with these recording technologies is that even when we put a probe into the brain and we keep it in the same place and we come back a few days later and we ask, okay, how has that neural representation changed due to the way that we process this data and the fact that we have to find the individual neurons from an aggregate recording, what could happen is that, we're looking at the same overall neural population, but one day we might be able to see just a subset of neurons, and then on another day we might see a different subset. So what this means is that even if there's some underlying dynamics that might be shared in this population, we're basically getting recordings which will come to us as multivariate time series data, but now the channels or the rows of this multivariate sequence could be flipped, could be permuted, or in the case of two different animals, there's actually no known correspondence between two different neurons in two different individuals' brains. So, and this has really been a kind of limiting factor in a lot of brain-machine interfaces, which try to learn some mapping between neural activity and behavior. But now that model, if you come back on a new day's worth of data, you have to recalibrate and you have to completely fine-tune that model again because of these shifts in the underlying distribution. So a few years ago now, we made a first attempt at building a transformer-based architecture that could deal with some of these underlying challenges, in particular the fact that you could get different numbers of neurons and not even the same neurons across different days, and of course across many animals where you have highly variable sets of underlying recordings. So I won't have time to go into the details of all of this, but I'll tell you about some of the key ingredients that we found were really essential in being able to scale up these types of models on electrophysiology data. The first thing is to, in the case where we actually have individual action potentials or spikes from individual neurons, our data will look a little bit like this. So we'll have different neurons that we've identified within the recording. And through the process of spike sorting, our goal is actually to figure out exactly when these action potentials or spikes are fired from a given neuron. So we can see actually within this electrophysiology data, it's very sparse, right? Neurons aren't constantly firing, and over the whole extent of time, we actually might have a small amount of underlying neural activity relative to the number of time steps, let's say, if we were to discretize. And so our main kind of contributions first was just figuring out how to tokenize this data. And so we do two things. The first is that we have a learnable neuron level embedding. So this kind of resembles a bit of what Michal was talking about, where we were talking about individual voxels being embedded within the model. Now every single neuron is going to have a learnable unit embedding that allows the model essentially over many different data sets to align these different neural recordings in some sort of functional way. So we can kind of think about that as, in language models, right? We could imagine that we have some sort of embedding of, dog and cat, where it places those two tokens close to each other and further away from pasta. And so we think about maybe a similar mechanism underlying the learning of these neuron level embeddings. And in particular, if I have two neurons that might have similar response profiles or similar functions, I imagine through this learnable unit embedding, I could actually place those two neurons close to one another. We did do some ablations in the work where we pass in neurons that it had seen previously, and indeed we do find that this mapping is sort of consistent. From the activity of these neurons, you can actually find these unit embeddings. And then the second piece is just instead of discretizing our spike train and representing all these zeros, we actually only pass in tokens for every single spike. And so now essentially a spike is given a unit embedding, which tells you what neuron it came from, and also we represent the timing information at which that spike was emitted. So this gives us a very sparse and very compressed representation, but one that actually has very high resolution in terms of the actual spiking dynamics. And then the second main ingredient is we adapted the perceiver IO architecture, which basically allows us to now take sequences of spikes from many different neurons, which could have really variable lengths. So we could have, 1,000 neurons or 10 neurons, and so those sequences will be very different in length. And so we use the perceiver, which basically uses cross-attention between the input sequence of spikes and a set of learnable latent tokens that are spread kind of uniformly along the context window. So now our latent tokens can basically take these variable length sequences and compress them into a length or a fixed length or fixed dimension. And then we apply multiple rounds of self-attention before reading out the final outputs. And there's different ways that we can train these models. This first version was actually going directly from neural activity to predict underlying behavioral sequences. And so this is, a supervised model in that sense, where we're going from neural activity to behavior. So in this first model, we showed that we could indeed use this type of architecture to scale up our models and also train on a lot of diverse data sets. This was the first attempt at actually training on different data sets from different laboratories. And so here we combined a little over 150 sessions worth of neural recordings from 9 non-human primates in both the premotor and primary motor cortex, while these animals were performing these 4 different movement tasks. So basically they would get a cue in this simple task, to move from the center towards one of 8 concentric targets. And then there's kind of more complex underlying movement distributions, like this random target task, where they have to move to a random target. Okay, so just to kind of summarize the results, so we found that overall, this ability to be able to train on more data was helpful at solving these underlying decoding tasks. In addition, we found that we could actually use data from different tasks and find that could actually improve the performance further. So this is just showing a summary of okay, what if I took the previous approach, which was to train a model on each session's worth of data and then look at the performance. So this is averaged over those 21 different single session models. Now what happens when I just combine all 21 of those sessions, so I get a significant improvement in the movement decoding performance, which we're measuring with the R² here. And then finally, what happens now if I add in 80 of these sessions from a different task? And so we do indeed find that having more data, even if it's from a different distribution or different task, can further benefit the decoding performance. And this can also be applied on new animals. So in this case, we can take the pre-trained model that's trained on however many sessions of neural recordings, and then on this new animal with a little bit of fine tuning, we can then achieve even better performance than the baseline model. So just to show that this pre-trained model can actually transfer on completely new individuals with just a small amount of fine tuning. It gets even crazier. So we've done a lot of work since then to like see the extent to which pre-training can actually transfer, even in scenarios where you have pretty different data distributions. How much does this potentially help? So in this example, this is recent work from NeurIPS last year. We showed that you can take a pre-trained model that's trained on all of these monkeys making different reaching movements, use that as your initialization point, and now actually transfer onto human motor cortical data from a patient that's paralyzed, that is not making any of those reaching movements, but is instead imagining writing different letters. So now our original task was to decode the motor outputs, but now we have a completely different task in humans, and indeed we do find that that pre-trained model is also helpful in this very different case, even transferring across different species. There's a lot of interesting open questions about what it's learning. And so it would be cool to talk with you all more about that. We've also done work in now looking at different self-supervised learning objectives. And in particular, we had some work where we looked at different forms of masking for the data and what are kind of most effective strategies that can be used to then transfer on different downstream tasks. Sorry. And in this particular work, we basically showed that perhaps in these kind of still limited data regimes, actually using many different types of masking strategies and predictive objectives can help, and they all kind of provide different sets of features to the model which are useful. And so in particular, there had been some previous work that had just looked at this co-smoothing approach, which is basically take a random subset of neurons, mask out their entire activity over some context window, and see if you can use the neighboring or other neural activity to be able to fill in those missing gaps. Also causal prediction, so being able to forecast and using all the prior neural data. But I would say, one thing that seems to be very useful in these types of cases where you actually have recordings from multiple different brain regions is to also use masking strategies that will force the model to use information across different brain regions in order to predict held out data. So something like this, where we do inter-region prediction, so using different brain regions to potentially predict held out activity. And this can be a really meaningful objective when we want to learn things about inter-region dynamics. And without this, the model can actually be quite myopic and use very local relationships. So, in this work, we have, just initially seen that there's been some significant improvements just due to the fact that you can train on multiple animals jointly, looking at the sort of scaling benefits as we increase the number of training animals. Okay, and then in the next part, or in the last part of my talk, I don't know how much longer do I have? Okay, 10 minutes, okay, great. So in the last part of the talk, I want to kind of slightly switch gears. These things are definitely interconnected, but to tell you about kind of another important ingredient that we've been working towards that fits into all of this. And that's namely that, in the model that I described before, we found that embedding every neuron and learning these unit level embeddings is really important for the ability to scale and to kind of unify these different data sets. And so we were really interested in whether or not we could learn these unit embeddings in a purely self-supervised way, because this could potentially be used as the vocabulary that's then fed into these foundation models or these other transformer models. And in particular, there's also just a body of literature in neuroscience that has identified kind of these core computational primitives or different cell types that exist in the brain. So, we like to treat in artificial neural networks like all neurons are created equal. They have similar activation functions, but we actually know that in the brain there are some core groups of specific cell types which have been typically annotated or found through transcriptomics and actually looking at their gene expression. And so there's a lot of interest in finding these cell types, identifying them, but it's a very challenging problem because due to the current tagging technologies, you can usually only get 1 or 2 cell types tagged in a given recording at a time. And so the goal of this is to really ask the question of, can we just from functional activity or just from looking at the dynamics of a population be able to identify some attributes of all of the individual neurons within that recording? And in particular, we have 2 key readouts. One is whether or not we can identify the cell type from those functional embeddings. And the other one is, could we also identify what brain region the neuron came from? And so to do that, like I've already kind of described, we'll take neural population activity, we'll pass this through some sort of encoder, and the goal would be that it's going to give us a channel level representation of all of the neurons that are passed through. And then from that, I could read out, the brain region or cell type. And so when designing, the objective and how this actual system will work, we had kind of 2 main design principles. The first is that so previous models would typically only look at like a single neuron's activity at a time and try to figure out what cell type it is. But so our first thing was just, okay, we should actually look at the population context, because knowing what my neighbors are doing should also inform me about what type of cell I am or where I'm from and what my function is. And so the first is finding a way to incorporate the population context. And then the second is just that we're going to use this assumption, which is, I think, a relatively loose one, which is that there is going to be some invariant, there's some property of that neuron that is stable over time. And so we'll use that just to design a contrastive loss that can encourage that kind of stability over different time points. So what do? We're going to take a long recording. We're going to select just randomly 2 different context windows from the recording, and then pass that through our encoder. We're going to use a set transformer without position encoding. So this allows us to actually pass in any population of neurons without any particular ordering, because that's a key thing that we would want to be able to do on a held out animal at test time. And so, so the first thing is we'll pass this chunk through our encoder. We do end up masking out some of the neurons so it doesn't learn just to only focus on one of its neighbors, and it can learn a more distributed relationship. And now we'll put these through the encoder, and really we're just going to use like a contrastive loss or something that looks like SimClear to bring the representation of the same neuron over different time points close in the latent space, and all other neurons I'll use as negative examples. So a pretty simple underlying objective, and we found, that this was actually a surprisingly robust and kind of generalizable loss that could be used on these types of data. We tested it under a number of different settings that varied in terms of the amount of generalization. The first being you can do the pre-training with the self-supervised loss on and training on some set of neurons from the same animals, both for your self-supervised as well as your linear layer that you're using to decode the brain region and cell type. This is a case where we can only use the testing animals in the unsupervised pre-training, but we don't get any examples of the cell type information from the test animals. And then this is the fully zero-shot mode, where we don't see any of the test animals in the self-supervised pre-training or in the training of the linear layer. So we tested this on a number of different data sets. The first is for cell type prediction, and the second being brain region classification. We found, this achieves state-of-the-art performance in many cases, like by a huge margin in comparison to the previous methods. A lot of that is just due to the fact that they didn't really have good ways of incorporating the population context. But also, because of the scale of this, we can also start to see a lot of benefits coming from pre-training on larger amounts of animals and larger amounts of data. So this is electrophysiology, and then we also applied it to calcium imaging data as well. In both cases, finding that we can actually get pretty significant zero-shot performance. And then we also did this for brain region prediction. All of this is those neuropixel probes that span multiple brain regions. What's kind of cool about this is that you have the same self-supervised pre-training and the same checkpoints, but then you can decode these two different pieces of information. So with those same representations, it has a lot of meaningful information about brain region and cell type. We also did some scaling analysis here, so just to show that, for instance, here, this is on the cell type prediction task that I talked about. This green curve is the pre-training only on 6 animals, 20 or 42. So we're actually seeing that for the cell type prediction, even with the largest data sets that we have available in this field, we're not yet saturating our performance, right? Like there's still some room to go. And so the work that we're doing now is to actually aggregate multiple data sets that all from the Allen Institute with different tasks, with some amount of this optotagging data to see, how far this can go. But we're really excited about the promise of this. And this is just to show that when we do scale up, we kind of see a lot of the session and animal-specific differences that are sort of present at smaller scale start to disappear. So we see more alignment of the representations across all of these distinct animals through this sort of, I guess you can imagine also seeing all these different animals experiencing the same task or same world kind of gives us these natural forms of augmentation. And so through scaling, we're actually seeing, more alignment of the latent representations. And so this is part of a larger research program and collaborative effort to, build foundation models. Now it's a bit of a buzzword, but we are very interested in being able to build these unified models, perhaps not only on electrophysiology data or calcium, but actually using multiple different neural modalities, all of which are presumably giving us different spatiotemporal resolutions, and so maybe unifying these different sources of data could also be helpful. We've already done a little bit of transfer across species, but I think that this could also be a program that could allow us to study evolutionary differences, or to understand what about perhaps the neural activity of macaques could we actually transfer to humans, and how do these different organisms compare, and and eventually combining many different types of tasks, visual and motor and memory. The downstream applications I've focused a lot on have been more brain-machine interfaces. So if you're interested in decoding from the brain, how does just seeing more brains and seeing more brains under diverse settings help your ability to stably decode from the brain? This has a lot of important implications in translational neuroscience and neurosurgery. We're interested and very excited about cell types because we think that if we can better functionally tag them, we'll be able to actually understand more about these motifs of computation in the brain, which are hard to look at now. And then, just having these pre-trained models that could be deployed in new settings as well. So I'll conclude there. I talked about kind of these different approaches that we've been taking towards scaling up and training on many diverse neural data sets for many different decoding applications. And then I was also happy to tell you a little bit about these efforts to try to learn embeddings that can give us this vocabulary of that can be used in new animals and in new settings. All right, thank you so much for your attention.
So before the learning method of neural identity was introduced, how did you traditionally map neural identity across individuals?
Yeah, great question. I kind of skimmed over that. So there's a few different approaches. All of them have their trade-offs and drawbacks. On one hand, if you want to do like in vivo recordings with electrophysiology, what people will do is something called optotagging. And so basically you have to start with like a transgenic mouse for which neurons of a specific type will express this light-sensitive protein, and then basically you just like go in and shine light on, and then those neurons that have that particular signal will fluoresce, and so you can detect them. So what that means is, I know it's very complicated, but basically because you have to have a transgenic mouse, it means only that type of cell can be tagged at once. And people are working on being able to have more than one color labeling strategy. So really it's like you do one animal, one type of cell you can label, and now you have another animal and a different cell type that you can label. So that's a very under-specified. And then the other case is you do a calcium imaging, so you don't get as much resolution of the temporal signatures, but then you'll basically sacrifice the animal and go in and you can do a gene expression map of all of the different neurons, both of which are very time consuming and costly. So we think if we could do this just directly on the functional activity, give all these neurons kind of these pseudo labels, then this could be like a scalable way to try to figure out these different circuits.
In the brain, you have all these connections between all the neurons, which I kind of didn't see how that enters in your model, except maybe in these representations that have some idea whom I also connected to.
Yes.
So in the neural identity, you have some additional information about connectivity. Is there any way to separate that in an interesting way? Or
Yeah,
sure, connectivity is part of neural identity. I understand that.
Right.
But it could also be interesting, to see maybe, or even discover connections that people have not really studied so far.
Yeah, no, I think that there's a lot of really exciting work where people are doing simultaneous recordings where you do activity, and you could also see some aspects of the connectivity. So that could be used perhaps as a way of confirming that these things are indeed, truly connected and all that. I would say that, disentangling the two is really hard. A lot of these objectives don't really get at this kind of like causal relationship between two different neurons, and so maybe to be able to resolve, are these two connected, you'll actually have to do some more like forecasting or something that really exerts this kind of more causal influence to identify the connections properly.
Deep data, maybe. You can connect that to some experimental setup and maybe test. I don't know.
Yes, so that's why I was saying there are some data sets where you have connections and neural activity, but it's very hard to get the connectivity. So it's always like these different trade-offs, and you're kind of putting together different pieces. But yes, in C. elegans and simple organisms, They have mapped out the connectome. So that's also another place where we could go. Yes.
Once you get out of primary sensory and motor areas, you have to deal with the fact that attention is actually changing the representation. So what is your strategy for dealing with that ugliness in this framework? Do you just essentially make your embedding the conjunction of every task and the selectivity in each of those tasks, or have you thought about that at all?
Yeah, in terms, so certainly once we get into higher level areas, also just our ability to relate that to some specific like decoding variable, et cetera, gets difficult. Yeah, I mean, I'm not entirely sure how to answer your question about like how we'll actually combine them once we have multiple different tasks in terms of concatenation or whatever. So yeah, sorry.
Okay. No problem.
Thank you
Yeah.
so much.
Okay, thanks.
Let's take a 1 hour break. We'll be back at 1:45 with you all. You don't want to miss it, so make sure you don't go too far.